In this video, we will discuss about the hypothesis that they checked is that exosome helps in the intercellular transfer of glycoRNAs, thereby helps in the intercellular communications through the transfer of glycoRNAs using exosomes. So in order to check that hypothesis, they designed their experimental plans that is shown in the panel A, where they took the cells and treat them with this AC4 GalNAc and then isolate the exosomes. And we have discussed that thing in our or discussed the results in our previous videos that these exosomes contains glycoRNAs. So what they did is they isolate the exosomes and incubate these isolated exosomes into the native HeLa cells. And in these native HeLa cells, uh, what they did is they check the cellular RNA contents of these native HeLa cells at different time intervals, that is from 1 to 20 hours. And this is what, and then check this RNA using this spec analysis uh, that can help detections of the glycoRNAs. So the panel B shows the result of these RNA isolations in the cellular HeLa cellular RNA isolations from this second culture. And here we can see that at different time intervals, that is at 0 time intervals, they do not see any glycoRNA presence, uh, whereas at after 1 hour treatment with these isolated exosomes from the first culture, they see a glycoRNA present in the cellular RNAs. And these things they have checked it till 20 hours and they can see the presence of this glycoRNAs in all the time conditions that they have tested: 1 hour, 15 hour, 1 hour, 5 hour, 10 hour, 20 hours. And in order to further confirm this their analysis, they check this glycoRNAs contains using the confocal macroscopy, and we can see that at the time of control they can see the presence of this glycoRNAs at the cellular membrane levels, but with time intervals, with different time intervals for the treatment of exosomes with this native HeLa incubation cells, they can see the glycoRNAs present in the intercellular parts of the cells that are mentioned here using the red dots at 1 hour time intervals, and at 5 hour time intervals also the number of this red dot increases, and these 10-hour time intervals, and at this 20-hour time intervals we can see the cellular glycoRNAs levels decreases significantly compared to the control, whereas the glycoRNAs signals within the cell membrane is increased. So this further substantiate their finding or their previous hypothesis that the exosomes helps in the intercellular transfer of these glycoRNAs. And in this case, we can see that these glycoRNAs, how they check their make sure that its glycoRNAs are formed in the exosomes. So for that they use this AC4 GalNAs, and they have checked its results in our previous videos where they have shown the experimental results that confirm that when the cells are treated with this AC GalNAc4, and the exosomes that are secreted by those cells will contain glycoRNAs. So this is how they make sure that the exosomes that are secreted from the first step contains glycoRNAs, whereas the native HeLa cells, since they are not treated with this AC GalNAs4, their exosomes, even though they contains the glycoRNAs, will not be stained by using this spec, because this spec will react with this AC4 GalNAs treated glycoRNAs that are formed in the exosomes. So only those exosomes, so only those this spec, only those glycoRNAs that are formed in the first step will be stained in this second culture, where the native HeLa cells are treated with the exosomes contains glycoRNAs from the first step. So the glycoRNAs that are formed by this native HeLa cells will not be detected using this spec method. This will ensure that whatever the glycoRNAs that are formed, or whatever the glycoRNAs that are detected in the second step are from the first steps. This is how they validated that exosomes helps in the intercellular transfer of this glycoRNAs. So I hope you understood how they validate their hypothesis. If you need to understand more about their experiments, it is how exosomes helps in the intercellular transfer of glycoRNAs. You can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.